Hello, my name is Craig Hodges from Case Western Reserve University. I'd first like to thank the moderators for the invitation to present in this symposium session. My talk will focus on using the mouse as a model to test CFTR-directed therapies. This talk will summarize the strengths and weaknesses of the mouse model, as well as highlight the use of novel mouse models that may assist the research community in testing CFTR-directed therapies in the future. I have no relationships to disclose related to this presentation. First, I would like to acknowledge that as a research community, we are very fortunate to have so many animal models to study cystic fibrosis and utilize them to test potential therapies. All of these are important models in their own right. I have drawn these arrows to represent that information is really a two-way street. Sometimes we start with the model and learn new information about cystic fibrosis or the CFTR gene and apply that to the patient. Other times we take information or clinical questions from the CF patient and study in the model. No matter which model is utilized, the ability to study CF and treatments in an in vivo model is quite important. A summary of each of these CF models can be found at cfanimalmodels.org, a website put together with the assistance of experts in each of these model systems. In this talk, I will focus on my favorite CF animal model, the mouse, and how it can be used to study CF-directed treatments. Some advantages of utilizing the mouse model in research in general are shown in this slide. While not all of these points are exclusive to the mouse, all of these points together make for a really strong model. The ability to control most conditions in genetically distinct background, the ability to produce many offspring in a short amount of time, the ability to easily manipulate a defined genome while having access to any tissue you desire, as well as the existence of a library of tools and strains available, and being space and cost efficient, all make the mouse a truly amazing animal model for research. Now, as far as the mouse as a model for cystic fibrosis, CF mice do show some level of involvement in almost every aspect of the CF disease in humans, ranging from reduced growth to infertility to inflammatory response. While late-stage lung disease is not observed in CF mice, they do not succumb to CF lung disease. Early lung disease manifestations similar to what is observed in CF patients are present. Exocrine pancreatic insufficiency is the one disease aspect that the mouse definitely does not recapitulate. There have been at least 16 different CF mouse models created over the years that have led to various insights into disease and have proved to be very useful in studying CF. I won't go into further detail of the specific disease phenotypes observed in the mouse or the differences between some of these various models, as this talk will focus on using the mouse specifically for testing CFTR-directed therapies. I will highlight some recent studies that fall under these three main categories: CFTR modulators, CFTR gene editing, and the potential for delivery of CFTR-directed therapies, all using the mouse as a model. First, I will discuss the use of CFTR modulators in the mouse. I show this figure from a recent review to summarize the components of certain FDA-approved CFTR modulators that will be discussed in this talk. Starting with a triple combination modulator in which two CFTR correctors, Trikafta and Alexacaftor, also known as VX661 and VX445, help F508del CFTR folding and increase its presence at the cell membrane, allowing for the potentiator, ivacaftor or VX770, to assist in opening CFTR to allow the movement of chloride and bicarbonate. These modulators were identified and developed without the use of any CF animal models, but there are still many questions about which aspects of CF are improved with these modulators and their long-term effects that may be easier to answer in animal models, such as the mouse. However, one of the most prevalent questions I hear about using mice in CF research is if these modulators work on mouse CFTR. The answer? It's complicated, but I'll do my best to summarize some of the work in the field. The first point to make is that human and mouse CFTR are not identical. On the gene level, they have a nucleotide sequence that's 82% identical in the coding region. And on the amino acid standpoint, human and mouse CFTR are 78% identical and 88% similar, meaning that the amino acids at a specific position might be of a similar size or charge, but the 12% are not the same amino acid. So is CFTR similar enough for modulators to be tested in the mouse? We start by investigating potentiators. In 2009, Van Goor and colleagues used the potentiator VX770 on wild-type mouse CFTR. In this figure, they measured forskolin-stimulated transepithelial current in oozing chamber experiments using wild-type mouse CFTR expressed in Fisher rat thyroid cells. They show a dose-response curve with genistein, shown here, which does potentiate mouse CFTR again. And then looking at VX770, it does not. The bar graph shows the average of several experiments. This suggests that VX770, which is a strong potentiator of human CFTR, did not potentiate mouse CFTR. Similarly, a more recent study showed wild-type mouse CFTR expressed in Chinese hamster ovary cells showed no potentiation with VX770 using patch clamp measurements. Here is where VX770 is added, and you would expect an increase in the curve if there was potentiation. Last, a study by Bose et al. Used an iodide efflux assay in CHO cells expressing not wild type, but F508del human or mouse CFTR. They observed a stronger response in human delta F with most potentiators. However, mouse did not show much potentiation. You can see that VX770 here showing a strong response in human, but no potentiation in mouse. And then you can see genistein here that shows strong potentiation in both. Interestingly, the same group used CFTR correctors as well and found that 15 correctors, including VX809, rescued both human and mouse F508del CFTR. However, most of these studies have been completed in overexpressing CFTR cells or cell lines rather than using primary cells from the mouse. My lab has been interested in CFTR modulators in the mouse using primary cells from the intestine that, when cultured, form intestinal organoids. These intestinal organoids contain crypt-like structures and an internal lumen lined with differentiated cells, recapitulating the in vivo tissue architecture. Intestinal CFTR is predominantly expressed in the apical membrane of the crypt cells, where its activation, in this instance forskolin, drives the secretion of electrolytes and fluids, leading to the increase in the size of the organoid. If CFTR is not functional, there's no increase in size can be observed. The use of these organoids to test CFTR function was really pioneered by Jeff Beekman's group in the Netherlands. Here's an example of wild-type intestinal organoids with forskolin added. You can see that almost all the organoids swell in a short period of time. We can measure how much all of these organoids swell in a field of view. And when you do that, you get a figure like this, where you can look at percent area normalized and see the different types of correctors or potentiators and the effect. And as you can see, using mouse F508del organoids, you can see that the correctors actually have quite an effect on swelling, but the potentiator VX770 does not. This is shown here, where the restoration, again, is seen with VX770 and 445, and even synergy between the two of them, whereas nothing is shown in VX770. So, do CFTR modulators work on mouse CFTR? I would say overall, while some potentiators do, most do not. Correctors have a similar responsiveness between mouse and human CFTR. But what about other modulators, such as read-through agents, as well as CFTR amplifiers and stabilizers? I think that may depend on the specific modulator, but I believe the mouse will be an important model to study these as well, because we need to know the long-term effects as well as what aspects of CF can be ameliorated by these modulators. Also, the ability to study these modulators throughout development, including in utero, can be studied thoroughly in the mouse. The next topic for utilizing the mouse as testing CFTR-directed therapies is gene editing. Remember, I mentioned that there is 82% sequence identity in the coding region of CFTR. Shown here is just exon 12 of human CFTR at the top and mouse CFTR exon 12 at the bottom. As you can see, the green and yellow changes are nucleotide base changes that are different but do not lead to amino acid changes. while the red and black base changes do lead to amino acid differences. As you can see, even these 11 differences would complicate things when trying to compare gene editing of human CFTR when using mouse CFTR. So to solve this problem, we made a human CFTR containing mouse model. This model was published last year and was a collaborative effort from many different groups. We accomplished this goal through the random insertion of the whole 188 kb CFTR, human CFTR gene, along with 40 kb upstream and 25 kb downstream. It was all contained in a bacterial artificial chromosome. To quickly summarize this model, the wild-type human CFTR is expressed in main tissues of interest in CF, displays function in the airway and intestine, and when put on a CF mouse background, meaning the mouse CFTR is non-functional, it corrects almost all CF mouse disease manifestations. This mouse by itself can be used to test gene editing of normal human CFTR gene sequence in an in vivo model, but since this publication, we have utilized the gene editing system CRISPR-Cas to create 5 new mutations. These 5 mutations, which include delta F508, G551D, and the 2 most common nonsense mutations, G542X and W282X, as well as a splicing mutation 3849 plus 10 kb C to T. They all contain the sequence found in human patients and should be able to be used for gene editing studies in vivo. But do these mutations impair CFTR function as you would expect in the mouse? And can they be corrected? I'll show you some data we have so far on these strains. We have assessed CFTR function in the airway of all 5 mutant strains using nasal potential difference assay. This assay is conducted in a live mouse and measures chloride secretion in the nasal lumen. In short, the more negative the output, the more CFTR function there is. Here you see a typical readout of the wild-type mouse, and next to it, the wild-type human CFTR expressing mouse, as well as the strains of mice that have the human CFTR with the different mutations. As you can see, wild-type human CFTR has function, but the 5 strains do not. If you use the same assay, but correct the mutation with CFTR modulators such as VX770 and G551D, VX774 G551D, or the triple combination for F508del, we have seen after 3 days of treatment, very strong correction for this nasal potential difference assay, as shown here. We have not had a chance to correct the 2 nonsense mutations or the splicing mutation, but we are working on those. In the intestine, in which we use intestinal sections from the mice in a nucing chamber and observe short circuit current before and after CFTR activation, we see once again that wild-type mouse shows robust CFTR activation. However, the wild-type human CFTR expressing mouse shows a reduced response, but clearly still CFTR function is evident. The mutations show little to no CFTR function. However, once again, we see the correction of G551D as well as delta F508 with modulators, suggesting these mutations are correctable in vivo. A similar response can be observed in vitro utilizing intestinal organoids from these mice as well. For instance, G551D human CFTR mice with no treatment, you see no swelling over time. However, when you treat with VX770, these intestinal organoids clearly show expansion, meaning that there is CFTR function. This data is tabulated here, in which you can observe both wild-type human CFTR as well as G551D are responsive to VX770. In F508del organoids from this mouse, CFTR is corrected by the triple combo drug. Again, no treatment, you see little to no expansion of the organoids, whereas treatment with triple combination, you see clear organoid CFTR function correction. This is tabulated here, showing correction using both correctors, VX661 with 770, as well as a triple combination. The advantages of the strains I just showed you are that they contain full-length human CFTR sequence and most regulatory sequence. They should be able to be used to investigate various gene editing tools and strategies, and they respond to CFTR modulators as well. However, there are some weaknesses to the models. When the original mouse was created, the back sequence inserted in multiple copies, so there are actually 6 copies of human CFTR in one location in the mouse genome. This may complicate data interpretation for certain gene editing strategies, like those that make double-stranded breaks. Also, we've observed that even multiple copies of human CFTR transgene, while it is expressed, have a lower level than mouse CFTR. Whether this is due to human and mouse transcription factor differences, we don't know yet. We are currently writing up the data for publication on all of these strains, but we are happy to share them and they are available for use now. Now let's move on to the last topic of delivery. I will specifically narrow the topic to delivery of gene editing components. This is a complicated topic, and there are very many different delivery strategies, as well as various cargoes to deliver. This is a great figure illustrating this point, in which either DNA or RNA is packaged in either viral vectors, liposomes, or peptides. And they are delivered to either cells from patients or delivered directly to the patients by inhalation or injection. The mouse has been a great model to test different delivery strategies, and while there are a vast number of studies using the mouse, I want to highlight a recent study that used the mouse to test the efficacy of delivering lipid nanoparticles. This work from Dan Siegwert's group from UT Southwestern Medical Center put their own spin on lipid nanoparticles. They use selective organ targeting, or what they term SORT, where LNPs are engineered to edit tissues via addition of supplemental SORT molecules, in this particular case, lipids. When used to introduce luciferase mRNA, they observed they can induce delivery to specific organs such as the lung, spleen, or liver. They also use the system to introduce gene editing materials that are used in CRISPR-Cas to remove stop cassettes in a genetic model, genetic mouse reporter model, in which the CRISPR-Cas components would remove the stop cassettes, allowing for TD tomato expression. They show that depending on the makeup of the SORT molecules they use, they could target the liver or the lung. This is an oversimplification of the work, but is a fantastic example of how the mouse can be used to test delivery strategies. While this published example is not specific to CF, I know that this group is using the system for gene editing and CFTR using mice. One tool that might assist the assessment of delivering gene editing components for the CFTR gene editing is displayed on the next slide. Here at Case Western Reserve University, Dr. Ron Conlon, Mitch Drum, and I are creating reporter mice that could be used to test such gene editing delivery strategies in the mouse for CFTR mutations. In this mouse, we have inserted human exon 11 and 12 sequence that's constitutively expressed throughout the mouse. We have inserted a nonsense mutation, in this case R553X. The idea is that if the mutation is corrected by gene editing or even read-through, you would get expression of a fluorescence gene, M. venus, shown here in yellow, and a luminescence gene, acoluciferase, shown here in red. This will allow the tracking of the mutation correction for assessing when, where, and how much correction occurred in the mouse. This can be done at specific time points in using fluorescence expression, or even throughout the life of a mouse using bioluminescence expression. We are creating not only the R553X, but G542X, W1282X using exon 23, as well as wild-type versions of these mice. This kind of information we are hoping to gather, the kind of information we are hoping to gather with these models is displayed on the next slide. While these are not examples of the mice we are creating, they do show how easy it is to use the mouse to identify fluorescent signaling. Here, we're using the red-green recombinase reporter mice, which reports the presence or absence of the enzyme Cre recombinase. Red is the absence, and green is the presence of Cre. We cryopreserved the whole mouse and then acquired high-resolution images using cryofluorescence imaging. This cryomicrodrome acquires whole animal high-resolution fluorescence and bright field images down to 2 micron level and provides fully automated cryosection and image reconstruction. Here we show Cre presence in the liver, intestine, and central nervous system. When put together and put in a 3D model, you can get a nice movie shown here where just Cre is expressed in the intestine. We are hoping to get this similar data from our CFTR gene editing reporter models, but more on a cell-by-cell basis. In conclusion, I hope I have provided you with an overview of how the mouse can be used to test CFTR-directed therapies when it comes to these three main topics: CFTR modulators, gene editing, and delivery. I would like to acknowledge all the people involved in the research presented from CASE, and if you are interested in any of these mice in the talk today, please contact me or find out more about them at these websites. I look forward to your questions in the live panel that is scheduled at the end of the symposium. Thank you for listening.
Hello, everybody. Thank you for joining the session today, and I'd like to thank the moderators for letting me give a talk on the CF rat as a model for advancing novel therapeutics. And I have no relationships to disclose. The rat as a species offers a number of advantages that enables its use as a model of CF disease. The relatively small size enables both age-related and long-term studies. It enables the testing of various therapeutic candidates, and it also enables live imaging and other physiologic metrics as outcome measures. The rat is a traditional model for physiologic studies in many organs, including the lung, and therefore there are an abundance of reagents, antibodies, and other molecular tools to enable assessment of disease in rat organs. The rat is also the traditional model for pharmacology and toxicology studies, and this lets the investigator move easily between preclinical experiments and the pharm tox that's necessary to advance a therapeutic forward. And the lower cost relative to the larger animal models allow for higher numbers in order to get your experiments done. So the first model I'm going to discuss today is the CFTR knockout. In order to create this model, we engineered a 16 base pair deletion in exon 3 of the rat sequence, generating a series of stop codons in exons 4 and 5. This results in deletion of the CFTR protein from all the tissues. Although I'm going to be focusing on the airway disease for the purposes of this talk, it's important to note that the CF model of rat does develop extrapulmonary organ manifestations. But when we look at the large airway, comparing the wild type to the knockout at 1 month of age, we see very little difference between the genotypes. However, by 6 months of age, now we start to see mucus impactions in these submucosal gland ducts denoted by the black arrows in the knockout that's absent in the wild type. And when we analyze these images morphometrically, we see that compared to the wild type, the knockout has smaller or the same size of submucosal glands at 1 and 3 months, which become hyperplagic by the age of 6 months. Correspondingly, intracellular mucus staining yields the same or smaller area in the knockout at 1 and 3 months compared to the wild type, while by 6 months there's more intracellular mucus staining in the large airways of the knockout. These are important factors in CF airway disease, and the fact that they develop progressively allows for studies into the natural history of CF lung disease. We also wanted to know if the mucus that's developed as the rat ages is abnormal. So we measure mucociliary transport and effective viscosity in situ on the large airways. At 1 month of age, again, you see that mucociliary transport is very normal in the knockout compared to the wild type. It starts to become more variable at 3 months of age, and by 6 months, the mucus is very static compared to the wild type. Similarly, the effective viscosity is the same in the knockout as the wild type at 1 month of age and starts to become more abnormal at 3 months of age, with a statistically significant higher viscosity at 6 months of age. So the progressive development of glandular hyperplasia also corresponds to increased mucus viscosity and decreased mucus transport. We wanted to know if this mucus would impact the susceptibility to infection. So we infected rats at 2 and 6 months of age. The 2-month-old rats, which have relatively normal mucus before infection, generate the same number of CFUs post-sacrifice in the wild type versus the knockout at 3 days post-infection. And both genotypes are able to clear the bacteria by 7 days after infection. In contrast, when the rats are infected at 6 months of age, the wild type grows back significant CFUs from the lungs collected at day 3. But by day 7, the wild type has cleared most of the bacteria from the lungs. The knockout, however, has higher CFUs at day 3, which drop initially at day 7, but then slowly climb back up to inoculum levels at day 28. So the abnormal viscosity of the mucus and the abnormal transport seen at 6 months of age permits chronic Pseudomonas infection. And we wanted to know if the infection correspondingly makes the mucus defect worse. So we looked at 2-month-old wild-type and knockout rats, stained the histology sections by ABPAS to draw out the mucus. At day 0 before infection, you can see that the wild-type and the knockout, again, are very similar by histology. By day 7, both genotypes are expressing more mucus in the airway, perhaps a little bit more in the knockout. But by day 28, again, both genotypes have completely cleared both the bacteria and the mucus from their airways. In contrast, at 6 months of age, the knockout starts with more mucus in the airways. You can see lining the epithelial surface in several small airways. At day 7 post-infection, again, both genotypes are making an abundance of mucus in the small airways, but you can see in the knockout these mucus strands that are stuck to the epithelial surface and unable to clear. This leads to a complete impaction of small airways with mucus by day 28 post-infection, whereas the wild-type airways have become clear again. So the 2-month-old knockout is able to resolve mucus accumulation following infection, but the 6-month-old knockout develops impacted airways by 28 days post-infection. So now we have a model that recapitulates several aspects of human airway disease. But what we cannot do with the CFTR knockout is study the effect of CFTR modulators. So with this in mind, we've generated a series of new mutations expressed in the rat. The first one I'm going to discuss is the generation of the F508 Del rat made in South Australia. This rat was generated by CRISPR-Cas9 gene editing. And when you look at the weights of the male and female rats, you can see the wild type in red are largest in both male and female, whereas the knockout in blue are significantly smaller, again, in both sexes. The F508 model comes in the middle with an intermediate weight phenotype. This is recapitulated in their survival data. As we've seen before, the knockout rat has significantly impaired survival, dropping below 50% by 40 days after birth. The F508, however, in the dashed line has higher survival, although still significantly reduced compared to the wild type. So the F508 rat develops an intermediate weight and survival phenotype. This group also wanted to see how CFTR was expressed in the smaller airways. So they studied bronchi and small airway from wild-type 508 and knockout rats and looked at mRNA expression using RNAscope. So in these experiments, the red dots indicate CFTR mRNA. You can see in the wild type in both the bronchi and the small airway, there are an abundance of red dots indicating expression. And when you zoom in, you can see multiple red dots per cell in the wild type. These number of red dots are diminished in the 508 with very little accumulation in the zoom in view, and in the knockout, they're almost completely absent. When this was quantified, they found that the F508 has a reduced number of dots compared to the wild type, although the knockout is significantly lower. And as a function of wild type, the 508 generates about 60% of CFTR mRNA compared to about 25% in the knockout. So the F508 Del rat expresses more CFTR mRNA compared to the knockout. They also looked at protein expression in the airways. In this case, the immunohistochemistry indicates CFTR in green. You can see in the wild type, there's plenty of CFTR staining throughout the airway, whereas in the 508 Del, zoomed in, the intensity is less, similar to the knockout. When co-expressed with a lysosomal marker, we can see that the wild type CFTR is preferentially expressed at the apical surface of the airway, whereas the F508 and the knockout CFTR is primarily lysosomal. So this indicates that F508 Del rat expresses inappropriately localized protein, suggesting the type of misfolding that's seen in the human data. A second F508del rat has been generated in Europe, and this group assessed the effects of this rat in response to modulators. This was also generated by CRISPR-Cas9 editing. And when they took nasal primary epithelial cells and cultured them at air-liquid interface, they were able to assess the short circuit current of the wild type versus the F508. The wild type has a strong response to forskolin with a corresponding decrease in current after INH172, indicating CFTR specificity. When the F508 del cells were analyzed without any treatment in the black line, you can see a smaller forskolin response and a smaller INH172 response. When the cells were treated with a combination of VX770 and 809, CFTR activity was increased as evidenced by a higher response to forskolin and INH172. In panel C, you can see the dotted line indicates 50% of wild-type activity, and the F508 del cells without treatment fall in somewhere at 25% of wild-type activity. The combination of 809 and 770 is able to increase activity to close to 50% in the F508 del cells, indicated as well by INH172 response. So the primary nasal cell cultures from this animal do respond to VX770 and VX809 with about 50% of wild type activity. We also wanted to generate a model that responded to the human drug ivacaftor. Now, ivacaftor has difficulty activating rodent CFTR, so to combat this problem, we created a human CFTR cDNA sequence with the G551D mutation and inserted it into the rat genome at exon 2. This generates a hybrid rat-human protein that is expressed behind the endogenous rat promoter, preserving the first exon and intron to allow for appropriate processing. We did test this in the cell line first, and if you compare the human-only sequence in the black to the rat-human hybrid in the blue, you can see similar responses to forskolin, VX770, and CFTR INH172, indicating that the hybrid protein behaves as well as the human. So this sequence was sent to Horizon Discovery to generate guide RNAs, preserving the first exon and intron of the rat genome, and the model was created using zinc finger nuclease. In order to ensure that the humanized G551D was expressed appropriately in the airway, we collected lung tissue from wild-type HG551D and knockout rats and analyzed CFTR mRNA expression. When probed with rat-specific primers, we saw a significant decrease in CFTR expression in the G551D compared to the wild type, similar to the knockout. When the tissue was probed using human-specific primers, we saw an abundance of mRNA in the G551D compared to both the wild type and the knockout. And when we used a set of primers that would cross-react both the human and the rat sequence, we saw roughly similar gene expression in the G551D compared to the wild type. And we also checked protein expression, and compared to the wild type, the G551D expresses about the same amount of protein in the airway compared to the absent band in the knockout. So this shows that the humanized G551D rat primarily expresses human sequence mRNA, and that the protein is expressed in the lung at a similar level to the wild type. In order to check the function, we excised trachea and measured short circuit current. The representative tracing in black indicates the wild type, and you can see the expected forskolin response that's counteracted with the INH172 response, indicating a high level of CFTR activity. Correspondingly, the G551D in the blue has a very low baseline activity comparable to the knockout in the red. And when we start adding VX770, we can see this current start to increase. When we look at the summary data on the right, we can see that in the blue, the currents become statistically significant from the knockout at about 100 nanomolars of VX770. Again, indicating that the G551D has the appropriate low baseline function compared to the wild type, and that it's responsive to ivacaftor. We also wanted to ensure that this rat responds to systemic treatment with ivacaftor. And to do this, we treated rats with 30 milligram per kilogram VX-770 by oral gavage for 14 days beginning at weaning. The G551D rat without treatment expresses a very small response to chloride-free ringers on nasal potential difference, whereas the G551D rat treated with VX-770 has an expected increase in CFTR activity as seen by the change in potential difference after chloride-free ringers. The summary data indicates that this restores G551D activity to about 50% of wild type with very minimal function in the knockout. And in this model, we don't see a differential amiloride response in any of the genotypes. So as seen with tracheal short circuit current, ivacaftor restores about 50% of wild type activity in the G551D rat. We also wanted to know if the abnormal mucus defects seen previously are responsive to ivacaftor. So again, we treated the rats for 14 days systemically and then measured the mucus viscosity and transport on the trachea. The expected drop in mucociliary transport was present in the G551D treated with vehicle only, and the addition of VX-770 restored mucociliary transport, again about to 50% of that seen in wild type. Additionally, the effective viscosity of the mucus in the G551D trachea was much higher compared to the wild type as expected with the CF phenotype. With the addition of VX-770, the viscosity dropped back down to wild type levels. So the ivacaftor was able to normalize the abnormal mucus following 14 days of systemic therapy. And we also wanted to generate a rat that would express a stop codon in order to test new therapeutics for the more rare mutations. So we generated a G542X rat by CRISPR-Cas9. This engineers a G to T mutation, as seen here with the asterisks, in the 1624 position to regenerate the 542X. When we measured mRNA expression in the red in this rat compared to wild type, we again saw significantly lower levels. And when we looked at protein of the G542X compared to wild type, we saw very absent protein expression in the airway. Tracheal epithelial cells from this rat were cultured on ALI and subjected to short circuit current measurements. You can see the vehicle in the red bar has very little activity. When the cells are treated with the traditional read-through agent G418, there is an increase in activity. Compared to amikacin in the summary data, we can see that there's a moderate response with amikacin and a response on G418 to approximately 2% of wild type activity. So the G542X rat does enable assessment of prototypical read-through agents. So to conclude, the CFTR knockout rat develops hyperplagic airway submucosal glands followed by delayed mucus clearance and hyperviscous airway mucus. These features enable chronic Pseudomonas aeruginosa infection that shows up at 6 months of age and is primarily dependent on abnormal mucus. So this model can be used to study and develop mucolytic agents, antimicrobial agents, or CFTR gene restoration that are all mutation agnostic. In order to study mutation-specific therapies, we turn to our newer models. The F508 models express F508 mRNA and protein in the bronchi and appear to be misfolded. They generate the expected bioelectric phenotype and do respond to VX-809 and VX-770. The humanized G551D rat responds to ivacaftor in a similar way to patients with about 50% of restoration of CFTR activity. And this really provides a benchmark for which to assess new CFTR targeting therapies or gene replacement. And then the G542X rat provides an in vivo model to test putative read-through agents and could potentially be used for gene editing approaches. And I'd like to acknowledge the folks in my lab who have helped enable this work, as well as others in the UAB Research Center and collaborators at Emory and Wellman Center for Photomedicine. Thank you.
I'd like to thank the organizers for inviting me to present our work. I'm going to be discussing today the use of genetic ferret models to understand CFTR function and CF pathogenesis. These are my disclosures. We are lucky as a field to have 7 animal models of cystic fibrosis. Mammalian models all develop intestinal complications as a result of CF and very extensive pancreatic, hepatic, and lung disease. However, ferret and pig models are the only models to date that have been shown to spontaneously develop lung disease. Today, I'm going to be discussing the use of genetic ferret models to understand CFTR function in the lung. When considering the use of animal models to study cystic fibrosis, it's important to appreciate the differences in cell biology within the conducting airways. Essentially, there are 4 trophic units that have a unique composition of cell types in stem cells and express CFTR in unique cell types that mediate lung innate immunity and clearance. In the proximal airways, these are the cartilaginous airways, humans, ferrets, pigs, and sheep all contain submucosal glands, which are important structures in secretion of mucin and fluid into the airways for mucociliary clearance. These structures also express abundant CFTR. However, in mouse and rats, submucosal glands are located only within the proximal trachea of mice and throughout the trachea of rats. I'm going to be discussing today the importance of pulmonary ionocytes as well as submucosal glands and potential connections between these 2 structures and cell types that are important for mucociliary clearance. In the proximal airway, there are predominantly 5 different cell types listed here. Intermediate cells are an intermediate state of differentiation of a basal cell towards columnar cells, and goblet cells, ionocyte, and ciliated cells also exist in various abundance. Recent single-cell CFTR expression profiling has revealed that ionocytes on a per-cell basis express the most abundant amount of CFTR, with less abundant CFTR expressed in secretory cells, as well as basal cells faded to become secretory cells. Thus, CFTR expression within the airway is highly regulated within different cellular compartments that are also in different abundance. But ionocytes only compose approximately 1 to 2% of the surface proximal airway epithelium. However, they express approximately 65% of the CFTR mRNA in that region. Ionocytes are also abundant within submucosal gland ducts and the surrounding surface airway epithelium. So what are the function of pulmonary ionocytes, and how can animal models help to address this question? There's a tremendous amount studied on the specification of ionocytes in multiple species, but the Rajagopal lab has demonstrated that FOXI1 is required for differentiation of a basal cell towards an ionocyte. When FOXI1 is deleted, CFTR expression is reduced by 87% in mouse tracheal airway epithelial cultures. However, despite the reduction in CFTR mRNA, there's actually an increase in the amount of chloride and bicarbonate current within FOXI1 knockout airway epithelia. This current appears to be mediated by a non-CFTR channel, but has properties very similar to CFTR. Alternatively, CFTR activation within a specific cell type may be able to function at a higher level for chloride and bicarbonate current in mouse in the absence of ionocytes. So we were interested in addressing whether this biology was conserved in species with abundant submucosal glands. And so Feng Yuan, a postdoc in my lab, and Grace Gazer, a research intern, tackled this question. They created a FOXI1 knockout ferret and demonstrated that FOXI1 expression was indeed reduced. This is in the kidney that's abundant in ionocytes. In polarized tracheal airway epithelial cells from these animals, FOXI1 expression is significantly reduced. Other ionocyte markers, ASCL3, also reduced, and CFTR mRNA was reduced by about 50%, with little change in these other markers of ciliated cells and secretory cells. When these cells were interrogated for both bicarbonate and chloride currents, we saw that there was a significant decrease in chloride current in response to forskolin IVMX, inhibitable by GlyH101, and quantified down here in both bicarbonate-free and chloride-free solutions. There was a slight change in transepithelial resistance in the absence of bicarbonate under chloride conditions. So ionocytes influence anion current in polarized tracheal epithelial cells, and there appear to be species-specific differences with mice. So how can such an infrequent cell type mediate a significant portion of anion conductance? And this is just one hypothesis in which ionocytes are expressing very abundant CFTR, mediating both bicarbonate and chloride movement across the epithelium, less CFTR expressed in secretory cells. And if this is true and ionocytes secrete bicarbonate, this secretion will be important in regulating sodium absorption through ENaC, which has been shown to be pH sensitive due to protease activation. Now, important in this hypothesis is the fact that an infrequent cell type like ionocytes could be connected with other cell types through gap junction to collectively mediate more anion movement than the cell could do on its own. And if we look here in a polarized airway epithelial culture in an early stage of differentiation, we see ciliated cells in this ionocyte with long appendages touching approximately 10 different cells within this culture. Now here, we haven't marked, but basolateral channels, which can also be specified in a cell-specific way, will of course play important roles in mediating the membrane potential in any one particular cell type that is also important for ion flow across the epithelium. Importantly, lung pathology within FOXI1 knockout ferrets was evident in a subset of animals. Here we show one FOXI1 knockout ferret that demonstrated a collapsed lung air trapping. If we look at higher power here in this large airway, one can see folding in or collapse of these airways. And importantly, in comparison to wild type animals here, there was significant submucosal gland hypoplasia. We can see very few tubules as compared to the submucosal glands in this bronchial airway in wild type animals. Furthermore, mucin strands were attaching to the surface of the proximal airways in FOXI1 knockout animals that were absent in wild types. Another interesting feature of pathology included enlargement of alveoli in an emphysema-like fashion. Again, one sees hypoplasia in this particular animal of the submucosal glands and potentially even of the bronchial epithelium as compared to the wild type animal here. 2 of 4 knockout animals displayed this hypoplastic gland and enlargement of alveoli. So what new genetic ferret models can help to inform ionocyte functions in the lung? We've created 3 different genetic ferret models. The first is our lineage tracing models, a ROSA TG model that switches from tomato to EGFP upon expression of CRE, and a second model, FOXI1 CRE ERT2, in which we knocked in an internal ribosome entry site upstream of CRE ERT2 into the 3' untranslated of the FOXI1 gene. The third model is a conditional CFTR knockout model, which is now breeding with these above models to test in a cell-specific way how CFTR deletion in ionocytes affects airway function. Lineage tracing in these double transgenic animals demonstrates the tracing of cells, similar abundance to ionocyte in the testes, here in the trachea, in the bronchi, as well as in distal tubules of submucosal glands, which was somewhat surprising, as well as collecting ducts. Importantly, the expression of the lineage trace EGFP co-localized with FOXI1 at the mRNA level here using single molecule FISH, and CFTR is expressed within FOXI1 expressing cells within the trachea here, looking at co-localization by single molecule FISH. We've created in vitro air-liquid interface cultures from these animals and are using this to understand the processes that are involved in specification of ionocytes from basal cells. We can see here labeling of cilia with ionocytes here in green. And interestingly, at early stages of differentiation, one sees that the ionocytes which are forming, here marked in white, following hydroxytamoxifen treatment of ALI cultures that are actively differentiating, one sees these are very active cells, extruding appendages and retracting, connecting with other cells. What this exactly means in terms of their function is currently unclear, but we're using these in vitro model to try to dissect that. We're currently breeding the conditional CFTR knockout ferret to the FOXI1 IRES CRE to allow for the study of cell-specific ablation of CFTR. And this model, this conditional model, can also be used with other Cre drivers to understand the contributions of secretory cells and or ciliated cells to anion conductance. Other CFTR models that have been useful include the CFTR knockout. We recently generated G551D ferret. I want to talk a little bit about their pathology and how this led us to study other models, mucin deletion models. CFTR knockout ferrets develop mucus accumulation in the airways as well as the submucosal glands compared to wild type. And the G551D ferret model, when removed from VX770, developed similar pathology. However, G551D animals maintained on VX770 are free from airway obstruction. Importantly, activation in vitro and polarized airway cultures, we can recover about 75% of chloride-mediated short-circuit current in the presence of VX770. These models are being used to understand both the onset and acute development of disease following removal of VX770 and also the reversal of these processes by reactivating VX770 treatment. The CFTR knockout ferret model has been useful in understanding inflammatory components of the disease process in the CF lung. Here we're comparing CF animals reared on symptomatic antibiotics. This is antibiotics given when an animal stopped gaining weight, or animals that were treated continuously from birth with 3 different antibiotics. And what you can see here is that the lifespan is greatly increased if treated with antibiotics from birth, but also the fact that at terminal disease, the group 2 animals treated continuously had no bacteria in the lung as compared to those that were treated symptomatically. If one compares the proteome in the group 2 animals treated continuously with antibiotic quantitatively to create a ratio between the CF and its non-CF pair, one sees an increase in inflammatory mediators here as well as MUC5B. And importantly, even continuous treatment with antibiotics did not protect the lung from bronchiectasis, mucus occlusion, and significant amount of inflammation. And in the 2 groups, these correlated significantly between mucus scores and inflammation scores using a histological index. So there appear to be 2 independent mechanisms of disease that occur in the CF lung: defective innate immunity that leads to infection and inflammation, and mucus accumulation that leads to inflammation. So we asked, what are the early changes that occur following loss of CFTR using the G551D ferret model? And in this study, we had ferrets, we maintained them on antibiotics to remove the secondary effects of bacterial infection and collected bronchial alveolar lavage while the animals were maintained on VX770 and then 30 days after removing. Proteomics analysis using isobaric tags with the ratio of in the CF state divided by the non-CF state, which would be in the presence of the VX770, demonstrated significant amount of inflammatory mediators contained within neutrophil granules, upregulation of MUC5AC, and a downregulation of MUC5B. Pathway analysis also confirmed a significant amount of inflammation, and disease pathway functions also come up with many indicators of pulmonary disease and inflammation. If one compares the MUC5B and MUC5AC expression between 2 groups of ferrets, chronic disease and knockout with knockout state divided by the wild type state, and the acute changes here performed in G551D animals taken off VX770 and prior to being taken off VX770, one sees that in the chronic state, MUC5B dominates within the bronchoalveolar lavage fluid. In the acute state, MUC5B is reduced and MUC5AC is increased. And so we were interested whether these changes in the composition of mucin subtypes impact disease progression in the CF lung, and whether or not the cellular source of MUC5B and MUC5AC impacts clearance and promotes inflammation. And to summarize this hypothesis, in the normal state, MUC5AC predominates within surface airway goblet cells with much less MUC5B, where glands are the predominant source of MUC5B. And in the CF state, this switches where MUC5B is now dominating in the surface airway epithelium, and the gland secretion is significantly impaired. Studies by the Welsh group demonstrated that MUC5B secretion from submucosal glands, that strands intertwine with MUC5AC produced from the surface airway epithelium. And you can see here in the surface airway epithelium, MUC5AC expression is dominating, where the majority of MUC5B is coming from glands. In the CF state, they've shown that MUC5B strands get stuck within the submucosal glands and can't adequately intertwine with the MUC5AC that's produced on the surface. Additionally, the MUC5B, you can see here, many more yellow cells and red cells are producing MUC5B, both alone and together with MUC5AC. In ferrets, we see that in non-CF ferrets, again, MUC5AC is predominantly expressed on the surface airway epithelium, MUC5B in the submucosal glands, and in CF state, there's significantly more MUC5B produced by the surface airway with MUC5AC. So to address the importance of MUC5B in mucociliary clearance, Nan He and Brad Rosen worked to characterize MUC5B knockout ferrets, and clearly there is impaired mucociliary clearance in MUC5B knockout ferrets, here tracheal en-face imaging with a fluorescent probe, as compared to wild-type animals, and that this is quantified as a rate. One sees a significant reduction in clearance rate. And also these animals had higher bacterial CFU within the bronchoalveolar lavage fluid. If one evaluates the mucin expressed from polarized tracheal airway epithelial cultures polarized in an air-liquid interface and compares them between wild-type and MUC5B knockout airway epithelia, one sees a lattice structure here in wild-type mucin that's not present in the MUC5B knockout, consistent with the potential hypotheses proposed by the Welsh group. And so we have a model for adaptation of mucin secretion and clearance following loss of CFTR, which in the normal state with good mucociliary clearance, the predominance of MUC5B in the proximal airways are coming from submucosal glands, intertwine in a lattice structure with MUC5AC produced by a different cell type that promotes good clearance. In the acute disease state, we hypothesize that the submucosal glands are just obstructed. They can no longer secrete, but there's been insufficient time for surface airway secretory cells to adapt to express MUC5B in an attempt to compensate. And in the adaptive state, we believe that the surface airway secretory cells or goblet cells in the proximal airway are producing both of these mucins, and this likely has functional implications for both clearance and perhaps inflammation within the airways that are reacting to the constant exposure to this mucin. We believe that ionocytes are important in this process since they're abundant within submucosal gland ducts and at the airway surface, and that the absence of function, CFTR function on ionocytes within glands likely affects the way that the mucins are folded on the airway surface and mediate clearance. We've shown that in CF, there's a reactive increase in ionocytes within the surface airway epithelium, likely promoting to attempt to compensate this process with secretion of MUC5B and MUC5AC from the same goblet cell. So to summarize, genetic ferret models can aid in the dissection of CFTR function at the cellular level. The conditional knockout, and I haven't discussed this, but reactivation of CFTR in a cell-specific fashion, I think will aid in dissecting unique functions of various cell types that express CFTR in the airway. The G5-5ND ferret model, we hope will be useful in dissecting the reversibility of some of these components of disease, and the MUC5B and MUC5AC ferret models on the CF background will assist in identifying how each of these mucins may contribute to disease progression in the absence of CFTR. We do hope that the G5-5ND ferret model will be adopted by other laboratories and facilitated by the fact that these animals can be easily reared now on CFTR potentiators. I'd like to thank those members of my group that have contributed to this work. This is really a team effort on nearly every type of project. I'd like to thank Brad Rosen and staff in his lab, Chuck Yeaman, a close collaborator on ionocytes, and Cal Parekh. Thank you.
Hello, I'm Patrick Sin from the University of Iowa. I'd like to thank the organizers for giving me the opportunity to talk today. Today I'll be talking about our work using AD, AAV, and lentivirus to deliver CFTR to CF pigs. Nothing to disclose. So very briefly, we're using the CF pig model. It's been around unbelievably for over 10 years. It's a large animal model that recapitulates CF human lung disease very well. It recapitulates the severe phenotype. Pig airways share similar anatomy with humans. And specifically today, the work I'm talking about, we use the gut-corrected CF pig. So it's a CF null pig that's transgenic for CFTR under the FAB promoter. So it expresses CFTR in the GI tract, but not in the lung. My talk today is divided into three main parts. First, I'll be talking about piggyback AAV. This was published in the Red Journal recently. And I'll talk about lentivirus, which was published in JC Insight, and then piggyback AD, which was in nucleic acid research. And there'll be a lot more details in these papers that I'm not going to talk about in my talk today. So piggyback AAV is a two-part system. Piggyback is a DNA transposon, and you can deliver it with a viral vector to basically make what's usually an episomal vector into an integrated vector. So in this case, the transposon is delivered with an AAV. So it's just your gene of interest, your expression cassette, and it's flanked by the piggyback TRs. So it's really just the addition of these TRs that make it a piggyback AAV. You deliver the transposase and trans, and it can be delivered in this schematic. It's with AD. It could be with another AAV. It could be with as an RNA, or it could be as a protein. So in the presence of transposase, the transposon is clipped out of the AAV and integrated into the genome. In the absence of transposase, this functions like any other AAV vector would. And we've shown in mice and in vitro that this system will stably integrate its cargo into the genome and persistently express for, in the case of a mouse, for the lifetime of a mouse. For the purposes of the talk today, in our studies to date in pigs, we've really only focused on the transposon component of this equation. So the stuff I'm going to talk about today, we didn't even supply transposase. We were more interested in establishing that this vector could in fact correct the CF phenotype in CF pigs. Getting CFTR into AAV is not terribly straightforward. We use the delta R construct, so it's human CFTR that lacks the R domain. So normally CFTR is about 4.5 kb. If you cut out the R domain, you save about 250 bases, which doesn't sound like a lot, but it's really important. We use the F5TG83 promoter, which is a very short promoter, and the minimal poly-A, which is 54 bases. These are described in the Engelhardt lab. We use the minimal piggyback TRs, which are combined, 5' and 3' are combined at 99 bases. So the whole insert was 4.6 kb, and if you add in the ITRs, it's about 4.9 kb. That really pushes the limit of what you can package into CFTR. So our goal was to just confirm that the CFTR was functional in a large animal model. So to deliver the AAV vector, this is basically our protocol. We recently published this in Biotechniques, if you're more interested in the details of our delivery protocol. But briefly, you anesthetize a newborn pig. These are one-day-old pigs that are about 1 kilo, roughly on average 1 kilo. They go down with light plain of anesthesia with isoflurane. You intubate with a 2.0 ET tube and then pass a pen century microsprayer through the ET tube. The tip of the microsprayer reaches about halfway down the trachea or so, and we deliver 10 to the 12th vector genomes, and it's formulated with doxorubicin. And we wait 2 weeks, give it enough time to express the transgene, and then we assay for phenotypic correction. We looked at multiple metrics, but the three I want to talk about today are ASL-pH, bacterial killing, and chloride current. And I'll be carrying this theme throughout my talk. We use the H22 capsid. So this is a pig-adapted capsid. It was just developed in Joe Zabner's lab, where they passaged an AAV library through live pig lungs and wound up with H22, which is probably most similar to 2, but has components of 5 and some other elements in it. So just immunostaining, if you look at a non-CF control, you get abundant CFTR expression. CF control, there's no CF. Pigs that receive the vector, there's a CFTR expression at the apical surface in ciliated cells. So we delivered the vector to pigs, to CF pigs, waited 2 weeks, and then we wanted to look to see if there was, in fact, evidence for chloride correction. So to that, we isolated tracheal explants directly from the pig and inserted them into oocing chambers. And this is just a picture of our setup. These are 8 different chambers that allow us to look at bioelectric properties of cells or tissue. So the explant was inserted into the chamber, and then you look for evidence of CFTR expression. So CF pigs have no chloride conductance in response to forskoliniamide, BMX, or GlyH. But in the pigs that received the vector, we saw a big response to F09 and GlyH. So this is reminiscent of, this is actually direct evidence of CFTR channel activity that approaches wild type levels. And we saw expression in both the trachea and the bronchus. We also looked at ASL pH, and so this is actually done in a live animal. So after you deliver the vector and you wait 2 weeks, you anesthetize the animal, you open a tracheal window, and you apply an opto directly to the ASL. CF pigs have, because of the bicarbonate transport, CF pigs have a lower tracheal ASL pH, and so their pH is around 7. Non-CF pigs have a pH of about 7.2, and in the CF pigs that received the vector, we saw restoration of the ASL pH back up to 7.2 in these newborn pigs. We looked at bacterial killing. So briefly, you coat a gold grid with bacteria, and again, just as the pH measurement, you open up a tracheal window in an anesthetized animal, you apply the grid directly to the ASL, and you wait 1 to 5 minutes. You take it off, you stain the bacteria for live dead markers, so the live cells are, live bacteria are green, the dead ones are red, and you just count the percentage of live cells. So CF pigs have very little bacterial killing. Non-CF pigs have bacterial killing of 70 to 80%, and then the pigs that receive the vector had complete restoration of the bacterial killing ability, back up to wild type levels. So these metrics suggest that we can in fact correct the CF phenotype at a short time point using an AAV-based vector. So moving on to lentivirus. For our lenti studies, we used an FIV-based vector, feline immunodeficiency virus. The transient construct itself was pretty bare bones. It had a codon-optimized pig CFTR and a SYN LTR. It was driven by the RSV promoter. It didn't have any really other bells and whistles, and that was on purpose. We wanted the vector to be, it was more important to us to get a good titer vector. So usually the smaller the vector, the better the titer when it comes to lentivirus. So titer was more important than expression off the construct. So we didn't really include any extra bells and whistles like the WPRE or the CPPT or some other things like that. And we used the GP64 envelope glycoprotein. This is from baculovirus, and we've published several papers showing that GP64 confers good airway tropism to the lentivirus. So we used this vector, we delivered it to pigs, and so we used a magic atomizer in this case instead of the microsprayer. We found that if you passed lentivirus through a microsprayer, it basically killed the titer. So the atomizer delivers slightly larger particles. It's a little, probably less pressure at the tip. It's a little gentler on the virus, and it had very little impact on the actual titer. So the atomizer is passed into the trachea, and similar to the AAV, we waited 2 weeks, and we looked at a lot of metrics that are in the paper that I described at the beginning, but Just for consistency's sakes, I'll talk about the leucine chamber data, the ASL-PH, and the bacterial killing again. So again, the trachea, an explant was taken directly from the trachea and put on the leucine chambers. Non-CF, you get a response to forced glutamine BMX of about 7 microamps. Response and CF, it's much lower, closer to 0. In the pigs that received the FIV CFTR, we saw restitution of the response to F9. It's similar with GlyH. You see a downward deflection with the channel blocker GlyH. The non-CF, it's about -7. CF, it's about 0. And the pigs that received vector, it looked very similar to the non-CF controls. Results weren't as impressive with the tracheal ASL-PH measurements in this case. The non-CF were about 7.2, the CF were about 7, and pigs that received the vector, there was a trend for that brought it back up on average, back up to 7.2, the same as the non-CF, but because of the variability, the p-value was only 0.28. Suggestion that there was a trend for improved ASL-PH that did not reach significance. We looked at bacterial killing, and as before, the non-CF is good at bacterial killing, the CF is not. And unfortunately, we only got measurements from two of the 3 animals. The third, when we were prepping to do the third animal, the bacteria didn't grow the previous day, which is a really cruddy hiccup to mess up a really expensive experiment. But of the 2 animals, on average, there was a trend for improved bacterial killing, where one was probably no different, the other one was about up to 50%. So it looked like we interpret this that as we were seeing some improvement in bacterial killing over what you get with CF. So now moving on to the piggyback add portion of the talk. So piggyback add, similar to piggyback AAV, is a 2-part system where the transposon is delivered in one add, and the transposase is delivered in a separate add, or AAV, or mRNA, or protein. In the absence of transposase, this construct functions like any other adenovirus. In the presence of transposase, the expression cassette is transposed from the ad genome and integrated into the host genome. And as before in mice, we've shown that this works quite well. You get expression out past a year in immunodeficient mice, and can correct, at least in vitro, can correct the CF phenotype. So in pigs, we picked a 5-day time point. So to date, we've only done an early time point. As before, we used the microsprayer, so we delivered the vector, either the transposon with the transposase or an cherry negative control to pigs, 10 to the 11th PFU, newborn pigs, and this was formulated with 0.1% LPC. That helps open up tight junctions and allows the vector access to basolateral receptor. After 5 days, we collect lungs. This is what a lung looks like. It has 6 lobes. Each lobe is cut up into smaller pieces. We looked at RNA and protein, or RNA and DNA, and all by qPCR, and we found widespread evidence of vector localization in all regions of all lungs of all animals. So you get really nice distribution based on PCR. Looking at immunofluorescence, so this is using a reporter gene, obviously GFP. We did immunostaining from sections all the way through the different lobes. And we quantified the size of each lumen. So we went from the large airways that have a lumen of about over 1 millimeter squared down to small airways that had lumens of 50 to 200 microns squared. And we counted GFP positive cells at the lumen. And we saw all the way down the tree GFP positive cells ranging from 34 to nearly 50%, which is just remarkably good levels of gene transfer efficiency in a large animal model. And perhaps more impressive, if you just take an entire lung and you dissect away the parenchyma, and take a picture of it under the dissecting scope. You can see really nice expression in all lobes, in large and small airways. So it's brighter in small airways because it's less tissue for the photons to permeate. But it is we do see expression, very widespread expression, which is pretty remarkable in my opinion. We looked at cell types. So the red is an alpha tubulin for cilia. We saw GFP positive cells in ciliated, non-ciliated, basal, secretory, and as well as submucosal glands. So we see it in all of the relevant airway types. A lot of these studies were done before ionocytes were even known, so we don't really do any staining for ionocytes, although to be honest, I think it would have been a real piece of luck to find expression in an ionocyte, which is based on the odds of finding an ionocyte in a section. I think that would have been pretty lucky to find even one. So now moving on to the CFTR delivery. So again, we took a tracheal explant and put it on into the oozing chamber. This is more of a close-up view of the chamber. CF pigs, there's no response to forced GLI BMX or GLI-H. Non-CF, you do get a response to F9 and GLI-H, and then the CF pigs that received the ad vector recapitulated the CFTR expression similar to the wild type levels. Similarly with the ASL-PH, CF pigs 7, non-CF about 7.2, and the pigs that received ad CFTR, pH was at or above what we saw with wild type animals, which is again quite remarkable. Bacterial killing, little in the CF, a lot in the non-CF, and the pigs that received ad CFTR restored it back up to non-CF levels. So this is just remarkable evidence by these metrics that we're getting really good gene transfer efficiency and phenotypic correction in these pigs, at least at an early time point, at least at 5 days. So next we look at actual protein expression. So this is these pictures were done by David Meyerholtz in the pathology core. The staining for CFTR, you can see it at the apical surface, similar to what you'd see with the GFP expression. If you zero in on an individual cell, so this animal received 2 ads, one that expressed GFP and one that expressed CFTR. You can see GFP expressed in the cytoplasm, and CFTR is tightly localized to the apical side. So there was some concern that overexpression of CFTR from an ad vector may cause mislocalization of the protein, but we do see nice tight expression at the apical surface. So just to conclude, I didn't show the data, but we've previously published data in mice. All of these vectors I talked about can confer persistent transgene expression in a mammal. And in pigs, we've demonstrated that AAV, lenti, and ad-based vectors can all be used to correct the anion channel defect in pigs. And in particular, AAV and ad were especially good at also doing at correcting the ASL-PH and bacterial killing ability. Importantly, we've demonstrated that aerosolized vectors can confer widespread gene transfer to the lungs of pigs. And that has really important implications for both for gene therapy, so both for gene addition, where you want to just supply a new cDNA, or gene correction, where you want to use CRISPR or base editors to go in and actually edit the endogenous locus. So these would be nice tools for delivering those kind of things. So future directions, we're currently working on investigating progenitor cells in particular. Ashley Cooney has a poster at this meeting about looking at our efforts, describing our efforts at looking at lentivirus in progenitor cells. And of course, we want to move past the short-term studies that I've talked about today and move more into longevity and safety studies in large animal models. So just briefly, I'm going to just go with some quick acknowledgements. I'd like to thank the funding sources. I won't name all the people who are involved and all the cores that are involved in these studies. It takes a lot of people to do these pig studies. I do want to point out Ashley Cooney, who is a postdoc in my lab, who did most of the stuff I talked about today, either hands-on, or she's the one who was the general contractor who made sure that all the things were running quite smoothly. She was a big asset. So with that, I will end it here, and I will thank you for your attention.